A very good morning everyone. So today I am presenting the journal titled Quorum in Streptococcus mutans regulates production of Tryglysin, a novel RaS RIPP antimicrobial compound. So this was published in the year 2021 in MBio, which is the American Society for Microbiology journal, which has an impact factor of 7. So quorum sensing is a bacterial communication through signaling molecules. So RAS RIPP is a radical enzyme, radical SAM enzyme. It modifies the ribosomally synthesized peptides, and Tryglysin is a novel antimicrobial peptide with tryptophan, glycine and lysine, which has a glycine linkage. And then bacteriocins are antimicrobial peptides produced by bacteria to compete with other species. Novelty is that it's the first study which reports a linkage between the Streptococcus mutans quorum sensing system and the Tryglysin production. So the structural elucidation of this Tryglysin A or B with a new RIPP subclass which has a unique tetrahydrobenzidor motif was put forth. And this studies also demonstrated the interspecies inhibition, suggesting the ecological role in oral microbiota competition. The aim of this study is to investigate the regulation and function of the WGK operon in Streptococcus mutans, this strain UA159 strain. The objective was to characterize the SHP RGB system and identify structurally analyze the triglycerides which are present and assess the antimicrobial activity against that particular streptococci. So the materials and methodology which they used is the bacterial strains and growth cultivation. They use primary strain for Streptococcus mutans. This strain, Streptococcus cereus of ESM 20646, which was there for comparative analysis, and they also used other Streptococcus species like Streptococcus pneumonia for inhibition analysis. And then they have this culture media. So there they use stored herbivore broth, which has a yeast extract in it which is used for rotating broth, and they also had a chemically defined medium CDM, which was also used for controlled experiments, and they also used E. coli of DL221 strain for heterologous expression of this triglycin. So the genetic manipulations were also done. So knockout mutants like alpha PDRA, which was used for RGG regulated deletion, and the one which had this biosynthetic component deletion was all taken into account. So they were constructed through the allelic replacement with spectinomycin resistant cassette. So that is how the knockout mutants were identified and manipulations were done. So reporter strains, these were the reporter strains taken for the promoter activity assays, and then complementation was done using this synthetic SHP peptide and reverse HSP peptide. So the synthetic one was the positive one and this reverse was taken as a negative control. And then they also used analytical techniques like RT sequence, qRT-PCR, mass spectrometry, like HPLC Q-TOF and was used. And then they also used phase contrast imaging to assess the shell signing post triglycid treatment. So they also made functional assays, like they tested for the antimicrobial activity using the broth curls with triglycins against the Streptococcus species. So the colony forming unit counts were used to distinguish the bacteriostatics versus the bactericidal effects, and also the UV inactivation was also done. So that confirmed the triglycin specific activity. And then statistical analysis like RNA sequence data, growth inhibition assays were done. So for growth inhibition assays, they used one-way ANOVA and SILAX post test for seeing the colony forming units per ml for comparison. And those response curves were also analyzed. And morphological quantification was also done. So that was the percentage of the chain cells were seen and analyzed through ANOVA. So those were the major statistical tests used. And then the results stated that there was a quorum sensing regulation that was observed in the Streptococcus mutant species. So this system activated the WGK operon transcription. So that the triglycin was also identified in that Streptococcus mutant species. So the triglycin A was seen in Streptococcus cereus species. So it was associated with this the gene with 4TA modification. Also, the triglycin B was seen in Streptococcus mutans species, but it had a cysteine substitution on the other hand. So that was a major difference which was identified. And then the antimicrobial activity stated that it inhibited the change, it inhibited the Streptococcus mutans, Streptococcus oralis, and Streptococcus pneumoniae. So there was a bacteriostatic effect which was observed on Streptococcus mutans species in general. So the morphological changes, it increased the cellular chaining among all the Streptococcus mutans species. Moving on to discussion. So the quorum sensing regulation. So this particular system, it activated the WGK opulon which linked the quorum sensing to the antimicrobial production. So it was very similar to that of Streptococcus pyogenes RGG systems, but with unique peptide specificity. And then the triglycine structure activity was also studied. So novel tetrahydrobenzenolone model was studied. And then cysteine substitution was also there. It was also seen in Streptococcus mutans species, but it did not alter the core activity. And then there was also an ecological role. So there was a very narrow spectrum inhibition which was seen in all the oral Streptococcus species, be it mitis or oralis, which suggested a competition between the species themselves. And the this inhibition, it implied the trade-offs which is seen in the bacteriocin production. So, but still this triglycin B was not observed in Streptococcus mutans, and mutants are supernates, so which possibly suggests degradation or lower expression of them. So there were also mechanistic gaps because there were no identified immunity genes which were seen, and there are certain unknown cellular targets of triglycins. And here in this study, no biofilm or animal model data was there to confirm or relate with the ecological impact. And then when compared to the previous literature, the study expanded beyond the antipeptides which was previously studied. And then there was QS bacteriocin link, as we saw before. So it parallels with that of the Streptococcus mutans system, but with a distinct regulatory target system. So the strength of this study is it has a multidisciplinary approach. So it combines genetics, biochemistry, and microbiology. And they have rigorously validated this particular study using RNA sequencing, qRT-PCR, multi-top, and also structural MS data. And ecological relevance is also there because it links the QS to the interspecies among themselves, the interspecies competition which is seen in the biofilms. And the limitation of the study is that the triglycin B is not detected in the Streptococcus mutans opponents. And there was an in vivo gap as said because no evidence of triglycin production in oral biofilms or animal models was seen. So that couldn't be correlated. And then the mechanism was actually unclear. The mode of action and immunity factors, they still remain unexplored. The future direction is that since that was not studied, they can do further in vivo studies that test the triglycin's role in particular in oral colonization and caries models. And for mechanistic insights, they can identify the cellular targets and immune mechanisms which were involved, and then put forth further results. And therapeutic potential is where they can engineer this triglycin for this selective anti-streptococcal agents, so which will be beneficial to prevent caries. So moving on to conclusion. So the specific implication is that this has a very novel antimicrobial class. So the triglycins, it represents a new RIPP subclass which has a greater potential for engineering them. So this also highlights quorum sensing as a driver of interspecies competition in oral biofilms. So this study it also has a very great translational potential where this could inspire narrow spectrum antibiotics targeting that pathogenetic streptococci, so by which can thereby prevent caries, and modifications may enhance stability or the activity of the species. So probiotic strategies can also be included because this engineered strains will control triglycin production to outcompete the caries pathogens itself. So the bottom line is this study elucidates a novel quorum sensing regulated antimicrobial system in mutants, highlighting this triglycin as a key player in despite all the technical limitations which were seen, so this study opens new avenues for exploring this RAS RIPP in microbiome modulation and antibiotic development. So this study, it had this 12 arrived guidelines, and it had a little bit of omics guidelines also because of the proteins and peptides involved. So it is a blend of all the guidelines, blend of 3 guidelines basically. The title and abstract, introduction, the overall here, the bacterial strains, the strains which were used, the genetic manipulation which was done, the analytical techniques which were used, mass spectrometry results, the functional assays, variables, data sources were all well documented. But then, in regard to the bias control, no explicit blinding for microscopy was done, and reverse HSP controls were used for negatives. That was partial. The statistical tests were all robust.